Hello, my name is Henry Magun. For the past 2 summers, I've worked at the Riverdale Summer Science Research Program under Dr. Cox, and the title of my project is The Characterization of Stress-Responding Protein Expression in the Heat-Shocked Marine Mollusk, a potential model for the response of acetylcholinesterase to heat. Now, first off, I'd like to give you a little introduction to acetylcholinesterase. Now, acetylcholine is a protein responsible for the transfer of signal from neuron to neuron. So here's acetylcholine, and you can see here's a neuron and here's a neuron, and acetylcholine is being transferred from one neuron to the next. And so here you can see acetylcholine is a neurotransmitter, and it transfers the signal from neuron to neuron. Acetylcholinesterase, on the other hand, is the enzyme responsible for breaking down acetylcholine, effectively stopping the transfer of the neuronal signal. Here's acetylcholinesterase breaking down acetylcholine into choline and acetate, therefore stopping, as I said, the transfer of the neuronal signal. Now, in the past, it has been thoroughly shown that the activity levels of acetylcholinesterase increase when an organism is introduced to pesticides. Because of this, acetylcholinesterase has become a commonly used biomarker by scientists to test the relative levels of stress an organism is under. This is actually the reason that we initially began using the protein, because our initial studies were a biomonitoring study of the Bronx River. However, as you can see, the increase of acetylcholinesterase activity due to heat shock has only been demonstrated twice, both times being in vitro, meaning within cell cultures, rather than in vivo, meaning in organisms. Now, for the past 2 years, we have noticed increases in the acetylcholinesterase levels of our heat-shocked mussels, and this is significant because it demonstrates the first evidence of an increase in acetylcholinesterase activity due to heat in vivo, meaning within the organisms. This led us to address the topic of the effect of heat on acetylcholinesterase. Now, the specific pathway in which heat affects acetylcholinesterase is largely unknown, and our goal is to identify and understand how stresses, specifically heat, affect acetylcholinesterase as a protein. Now, there are 4 possible models that we have provided for the effect of heat on acetylcholinesterase. The first model being heat shock directly affecting acetylcholinesterase activity. Now, enzymes catalyze chemical reactions, in this case, the breaking down of acetylcholine. All enzymes have optimal conditions in which they can most efficiently speed up their respective reactions. Heat shocking, therefore, may bring the muscles closer to the optimal temperature for acetylcholinesterase to perform its job, and therefore it would increase its activity. Now, model 2, heat shock directly affecting the expression of acetylcholinesterase. Now, as Maddie mentioned in his last talk, heat directly causes the expression of HSP70 via heat-responsive promoter sequences. It has been shown by numerous labs that such heat-responsive promoters are also present upstream of the acetylcholinesterase gene in an organism's genome. Therefore, as an organism is introduced to heat, the heat-responsive promoter sequences are activated, and everything downstream of them becomes transcribed. And if acetylcholinesterase is downstream of this promoter sequence, it is therefore transcribed, increasing its expression and therefore increasing its activity. Now, model 3, heat shock, so protein 70 acting as a chaperone protein to denatured acetylcholinesterase. Now, as Maddie also mentioned, heat shock protein 70 has the ability to renature denatured proteins. Now, due to heat shocking, the acetylcholinesterase may have been denatured. And therefore, an increase in HSP70 levels caused by the heat shock could renature the acetylcholinesterase that was previously denatured and cause an increase in activity. Model 4, HSP70 recruits inactive stores of acetylcholinesterase. Now, enzymes can be inactivated by protein inhibitors as well as reactivated by protein activators. It has been shown by numerous labs that HSP70 has the potential to activate certain inactive enzymes. Therefore, an increase in HSP70 expression caused by heat shock may activate certain inactive stores of acetylcholinesterase. In this image, the allosteric activator would be HSP70, and this would represent an inactive acetylcholinesterase enzyme. However, once HSP70 is bound to the acetylcholinesterase enzyme becomes active and is therefore allowed to perform its job, increasing its activity levels. Conclusions. Our study is ongoing as we are currently testing the HSP70 expression, acetylcholinesterase expression, and acetylcholinesterase activity levels of control and heat-shocked muscle samples over a time course. Such data, we hope, will allow us to determine which model for how heat affects acetylcholinesterase is appropriate. Now, I'd like to thank everyone who supported the Summer Science Research Program, especially the Marga Foundation for providing grant funding to aid our research. I'd also especially like to thank Dr. Cox, because without her, none of this would be possible, and all the Summer Science Research Program would not be possible. So thank you, Dr. Cox.